Welcome everybody to the Bioexcel webinar number 8. So now we start again the webinar series of Bioexcel, and we have a lot of speakers in particular in these first months of the year, January, February, March, April, and maybe also May. So keep an eye on what is going on. So our first webinar this winter-spring season, it's given by Attilio Vargiu. Attilio is from the University of Cagliari, and he will tell us about how to account for protein and ligand flexibility in molecular docking and in virtual screening. And so in particular, it's titled Accounting for Protein and Ligand Flexibility in Molecular Docking and Virtual Screening of Challenging Target. Just for your awareness, this webinar is recorded, and the recording will upload after on the Bioexcel webpage. During the webinar, you can ask questions using the function, Q&A function that you find at the bottom of the Zoom application. You might see this symbol, on this symbol, depending on which operating system you have. And so you just type that your question. It will be great if you can tell us if you have or not a microphone, because in case you have a microphone, we will just unmute you. So you have, if we have time, so you can ask yourself your question. So if you have question after the webinar, you are most welcome to join the Bioexcel forum, Ask Bioexcel. In particular, there is a category called Bioexcel Webinar, where you find a post dedicated to Attilio webinar. So you can ask extra question there, and Attilio will be very happy to answer to those questions. So now something about Attilio. So Attilio got a master at the University of Cagliari, and then he moved for his PhD in biological physics at CISA, that is International School for Advanced Study, and is located in Trieste, also in Italy. Then almost 11 years, between 2008 and 2019, he worked in a different country. So he worked in Italy, at the University of Cagliari, but also in Germany, at Jacobs University in Bremen, in the Netherlands at Utrecht University, and USA in the University of California, Berkeley. And finally, he settled down in 2019, where he got appointment as assistant professor for applied physics at the University of Cagliari, and now is associate professor in the same position. His research focuses on simulating all different kinds of molecular micromolecules, like peptides, but also small molecule, membrane, and also in particular is interested in their assembling, and use different type of computational method, going from quantum-based approach to coarse-grain and machine learning approaches. Currently, the main research line of Attilio focuses on bacterial resistance to antibiotics mediated by multi-drug transporter, self-assembly of chiral peptide, for technological biomedical application, and development of method and protocol for accurately predict the structure of challenging protein-ligand complex. And Attilio is also part of the Bioexcel Ambassador Program, and in particular, he represents Italy inside the Bioexcel Ambassador Program. So now I stop here and I give the word to Attilio. Please, Attilio.
Okay, thank you. Thank you very much, Alessandra. Thank you all for attending. As Alessandra said, I will now share my screen, and I will try to tell you about okay. I will try to put this down. Okay. I hope everyone can see the screen. Yeah, thank you again for the invitation. It's a pleasure to be invited to this webinar series. And today I will try to show you the results of a project that is running in our lab for about 6 years, in which we try to develop a protocol to account for protein ligand flexibility in molecular docking and virtual screening, in particular addressing the flexibility challenge. So, sorry. Okay, yes, here is the outline of my talk. I will give a very brief background, then I will present our method and a few applications. And then I will draw a few conclusions, and I will talk about the perspective of the work that I will be talking about. So, but before, I would like to tell you where we are located. We are located in Cagliari, that is in Sardinia, the island in the middle of the Tyrrhenian Sea, in the beautiful city of Cagliari. And this is our lab. Okay, so I guess you can recognize the computer room that is naturally cooled. And okay, jokes aside, let's come to the to the topic of the presentation. In particular, we addressed, as I said, the flexibility challenge in structure-based drug discovery. Because, as you know, proteins and ligands are very flexible entities, and their arrangement depends on the context. The same protein can interact in different ways with different partners, and in particular, many proteins assume a very different conformation when they are unbound with respect to the conformation they assume when they are bound to a partner. The partner can be another protein, DNA, other macromolecules in general, but also ligands. And we will be focusing on this. Accounting for the degrees of freedom that are involved in these conformational changes, of course, it's crucial to predict the complex, the stable structures formed by proteins and ligands, for instance, by molecular docking. Among the several ways that have been developed over the last decades to account for this, there is ensemble docking that consists in docking ligands on an ensemble of structures, okay, of a protein, because if you take different conformations, you can also account for different shapes and different volumes of the putative binding site where the ligand should bind, okay? And in this way, you can hope that you find the correct conformation assumed by the protein in the interaction with its substrate, for instance. So, of course, these structures can be found by experimental methods and by several computational methods. Okay. Our approach is one of the methods that have been developed in these years to try to reconstruct the structure of a protein interacting with a ligand, and is called gEDES, that stands for Generalized Ensemble Docking with Enhanced Sampling of Pocket Shape. I will briefly describe our methodology. I want to say from the beginning that we don't rely on any experimental information of the protein or similar proteins bound to any ligand. So we exploit the unbound structure, the apostructure of the protein. Then we use computational methods to detect the putative binding site or binding sites on the protein. And also we use other computational methods developed by other groups that identify the so-called quasi-rigid domains that move like a wall during the interaction between a protein and a ligand, or during the, let's say, change, if you want, between inactive and active conformation of an enzyme protein, for instance. Once we have this data, we use bias sampling methods that are based on collective variables. For those of you that are not familiar with the concept of collective variables, just briefly, I will say that they are parameters generally related to the structure, such as distances, angles, dihedrals, and even more complex ones, that you can bias in molecular dynamics simulation to enhance the conformational sampling of different structures. And in our methodology, we developed ad hoc general collective variables specifically tailored to sample apo to all conformational transitions. The first one is the radius of gyration of the binding site that you can use to shrink or expand the binding site itself. And then we develop the so-called contacts across inertial planes in the binding site, and the quasi-rigid, and the contacts across the quasi-rigid domains within the same binding site. What are these collective variables? Basically, we make a simple operation. We take the binding site, we calculate the principal axis of the binding site, and then we draw planes that pass through the geometrical center of the binding site and are orthogonal to each principal axis. In this way, you can divide the amino acids of the binding site into groups that are shown here in green and orange on the left and on the right side, on opposite sides of each inertia plane. Then, if you do this automatic division of the binding site residues into two groups, you can sample the conformation of the binding site by applying a bias on the number of pseudo contacts between these two groups. Okay, since you have three such planes, you bias on three independent collective variables, and in this way, you effectively sample different shapes and different volumes of the binding site. What happens if you have a binding site that is in between two or more quasi-rigid domains? You can mimic hinge-like motions, for instance, by defining additional planes that are shown in this picture in green and in yellow, for instance, between the three quasi-rigid domains of the adenylate kinase protein, which I will return later. And in this case, you can reproduce functional motions associated to activation, inactivation, or substrate binding to the protein. Finally, in the case where you have a large percentage of charged amino acids in the binding site above a threshold that we generally set to 20 or 25%, you can also exploit additional collective variables. Why? Because if you have a lot of charged amino acids, this conformation that you see here, in which we have two groups on opposite sides of the green plane, could have charged amino acids placed in the position that you see in the picture. But from the point of view of calculating the pseudo contacts between these two groups, the two conformations that you see in these slides are perfectly equivalent at the level of functional interactions, because in this case, if you have a charged ligand that should bind to this protein, in this case, you don't reproduce, in the case on the left, you don't reproduce the protein ligand interactions. In the case in the right, you have all the charged ligands that interact with the charged residues that interact with the charged ligand, and this is the conformation you want to obtain. So how to do this? We used a very simple approach. We basically split the residues into groups. So in one group, you will take only the non-charged amino acids. In the other group, we take only the charged amino acids. In this way, we force conformational change among the charged amino acids that generally interact more strongly, and we experienced in our research that they do not move as they should in order to orient in the proper way to interact with the ligands. So if you have a protein like this, the apoconformation, you have a large distance between charged amino acids on two sides of the protein. In the holo conformation, these distances are very, very short. And if you are able to reproduce this conformation, then you can reproduce the correct interaction between this protein and its substrate or substrates. So once we have defined this set of collective variables, we can apply metadynamics. That is a technique that I'm sure most of you know, that is based on a history-dependent bias potential that you add to the system in order to enhance the sampling of different states. In particular, in this case, this state could be the apo state of the protein, and this state could be the holo state of the protein. Then once you perform metadynamic simulations that have the advantage that you don't need to know the structure of the complex. So you start from the apoconformation of the protein, and you hope that during your sampling, you also sample the holo-like conformations of the protein. Then we perform a multi-step cluster analysis in the collective variable space. In particular, we apply restraints because if this is the radius of gyration of the binding site, we notice that often the most interesting conformations are located either on one or the other tail of the distribution. Why? Because in general, you start from the apostructure, so most of the structure that you will sample are still very close to the apo1, and the bound ones are either at the left or at the right side of the distribution, at least for very flexible proteins. So we want a distribution like this one. And how do we achieve this? We use a very simple recipe in which we first perform a hierarchical clustering, and then we feed another method, k-means, with the initial structure taken from hierarchical clustering and we apply restraints in order to have a given number of structures corresponding to each value of the radius of gyration that was sampled during our metadynamic simulations. You see in this graph that if you compare the distribution that you sample during metadynamics simulation and the cluster that you get by performing only k-means with random initial structures, if we apply our methodology, we increase, you see, the percentage of conformations associated to very low or very high values of the radius of gyration. And in this specific case, these conformations are holo-like conformations. So we increase the possibility to sample holo-like conformations. And then we can use these structures, for instance, as I said, in molecular docking, in vertical screening, in also additional applications. And in particular, in the rest of my talk, I will show you data obtained with using two docking softwares that are very well known in the community, Autodock and Undock, that use a different philosophy. We wanted to test two protocols in order to verify the robustness of our approach against different docking protocols. So to validate the method, we started taking a set of 3 proteins that are very different with respect to the level of conformational change associated to ligand binding. The three proteins are shown here are recombinant ricin A hydrolase, a beta-glucosyl transferase, and the allose binding protein. You see that the first one undergo a very small conformational change, just a shift in a tyrosine corresponding to a change in the binding site of only 1 angstrom. The second one, it's more relevant. There are motions at the level of the domains of the protein, and you see a lot of residues that change conformation, almost 3 angstrom. And the allose binding protein, it's even more flexible. Okay, you see a lot of residues changing conformation during the binding of a sugar molecule. And so the idea was to see how our approach perform with different targets. So it is general enough to be applied to all of them. Here I show you first the sampling and the docking results. Regarding the sampling, here I show the fraction of bound-like, holo-like conformations sampled at the binding site, and I compare our simulations with standard molecular dynamic simulations of 10 microseconds in length. Our gEDES simulations are about 500, 600 nanoseconds in length instead. You see here the percentage of conformations that have an RMSD from the true holo structure below 2 angstrom. Note that this RMSD is calculated not at the level of the C alpha, but at the level of the all heavy atoms of the binding site. You see that in the case of the very rigid protein ricin, the standard molecular dynamics and our approach perform equally well, and this was expected. But if we go to the other 2 targets, you see that for both of them, only our approach was able to generate a small but still relevant fraction of conformations that can be classified as holo-like. And in parentheses, you see the minimum RMSD that we were able to generate with respect to the conformation in the true complex. So how does this reflect on the docking performance? Here I show you the native-like poses obtained using both autodock and for and addock. You see again that for rising, both approaches perform very well. And again, in the case of beta-glucosyl transferase and allose binding protein, instead, in particular with the last one, only our approach was able to find the correct pose and rank the correct pose among the top 10, for sure, even top 2 in this case. Okay, then once we validated the method, we decided to perform a more challenging test, in particular to use our approach in the grand challenge. That is, before I have to say something. Here you see the main reason why we think our approach is effective. Here in gray, you see the true structure of the complex between the ligand that binds to beta-glucosyl transferase and the position of the residues that are binding to the ligand. And in red, you see the best structure that we were able to generate by standard molecular dynamic simulations. You see that there is one amino acid that is in the middle of the binding site, so the ligand will never bind in the proper position. Our approach, in contrast, was able to generate this kind of structure. You see that it's not perfect, of course, but it still has a lot, the majority actually of amino acid simultaneously assuming the right conformation. And this allows the ligand to bind within the binding site. We like to let's say, compare the first approach to a watermelon that where you have a bigger binding site compared to the experimental one, but this is filled. In the second case, we have a coconut where we compress the binding site, but at the same time, we create a void inside. And this is achieved by using our ad hoc collective variables. Then, as I said, we participated to the Grand Challenge 4 in 2020. And in this challenge, we were asked to determine the native-like poses of 20 ligands on the beta-secretase protein, where we were given only the sequence of the protein and the smiles of 20 ligands. We were allowed to upload only 5 poses per ligand. And of course, we didn't know anything about the experimental structure that was released after the competition was closed. So in this case, we prepared the ligands by calculating the most likely protonation state at physiological pH. We generate conformers of the ligand using one of the tools that were available, in particular OpenEye Omega. And we used in this case, only for the ligands, an approach, a template-based approach in which we took the 10 most likely conformers by comparison with structure that were available in the database. We generate instead the conformation of the protein using our gEDES approach, and then we performed docking, again with ad hoc and with autodock. There were more than 100 groups that participated to this challenge. And in this graph, you see the average minimum root mean square deviation obtained by all the groups. You see that there were groups performing very well with sub-angstrom RMSD, but we also performed very well. We got RMSD below 2 with autodock and close to 2 with addock. And we reproduced correctly the native poses for 18 out of 20 ligands. Note that we were the only group in this competition that didn't use any information about the experimental structure of the protein when deriving the models used in docking of the 20 ligands. Okay, then we assessed very challenging targets. That is adenylate kinase. That is an enzyme allosteric and endowed with multiple multiple binding sites in the same location of the protein. This protein is made by 3 domains, as you can see here, the gray one that is called the core domain, the ATP binding domain called the lead in green, and the NP binding domain in yellow. Okay, you see that there is a very large conformational change upon binding of ligands to this protein. Particularly here, you see the binding of an inhibitor to the protein that correspond to an RMSD change of about 7 angstrom concerning the whole protein and 5 angstrom concerning the binding site. The binding site is very large in this case. There are a lot of studies that use this protein because it's very flexible to benchmark protocols to generate holo-like conformations of proteins. So here you see a movie in which I hope it's clear that we can, by sampling only the region of the binding site with our approach on the right, you see in liquorized binding site and in yellow the reference, drag the full protein to assume the holo-like conformation. You see that we end up with a structure of the binding site that is very close to the holo conformation, and this was obtained with a structure of the protein that was also very close to the holo conformation. Note that this movie does not represent an MD trajectory. I just took the clusters that we extracted from our gEDES simulation and I sorted them according to the RMSD from the experimental conformation to show that there is this coupling between the conformation of the full protein and the conformation of the ligand and the conformation of the binding site. Okay, then in this case, we tested our approach for the ability to reproduce the conformations of several complexes. An inhibitor that binds to both the lead core and NMP core interfaces, and the protein assumes a closed form. Then ADP that binds to the lead core interface, AMP, adenosine monophosphate that binds to the NMP core interface, and this is very interesting to us, a catalytically incompetent GTP analog that binds also to the lead core, but in the open, inactive conformation of the protein. So here I report the results of the sampling. Again, you see on the top in gray and black, the results obtained with gEDES, and in green, the results obtained by standard molecular dynamics simulations. And you see in both cases the fraction on the y-axis, the fraction of conformations assuming a given value of the RMSD with respect to the experimental conformation. You see here that you have, regarding the inhibitor AP5, a very good shift obtained by gEDES that is able to reproduce conformation almost having almost an RMSD of almost 2 angstrom with respect to the holo conformation, while the standard MD was unable to obtain any relevant conformation. And here I show you the best conformation that we obtained for this site. And in particular, you see here again in green and in black, the fraction of charged residues that simultaneously assume a native conformation and a fraction of non-charged residues that also assume a native conformation. You see that in particular here, with gEDES, we were able to obtain more than 90% of the charged residues assuming a native-like conformation, and 75% almost of the non-charged residues also assuming holo-like conformation. While in the case of standard MD, you see we have a problem in both cases because we were able to reproduce a very small fraction of amino acids in the correct orientation. So we calculate the same distribution for all the binding sites, and you see that in all cases, but of course, in the last one, where I recall you that the conformation of the protein is open, the two approaches were more or less equivalent. Again, when we go to use these conformations in molecular docking, we were able to reproduce among the top 10 conformations the binding of the inhibitor, the binding of the ADP ligand, AMP ligand, and moreover, the binding of the GCP ligand. Binding obtained on a conformation, as you can see here, where in dark yellow and dark colors in general, we have the experimental structure, and in bright colors, we have the best conformation generated with our approach. You see, of course, they are not perfectly superposable, and this was expected because our approach does not use any experimental information on the complex, but still it was good enough to allow for a good binding, to retrieve a good binding pose of the ligand. And then we compare this approach with deep learning-based tools. You see in this table a report for each ligand and for each approach, autodoc coupled with gEDES, addoc coupled with gEDES, and neural plexer, that is a recent tool that is able to generate conformations of protein ligand complexes exploiting artificial intelligence methods. Here you see the RMSD of the ligand in the best pose end of the binding site and the ranking of the pose obtained. You see that the results are comparable between the two, although in the first two cases, neural plexer performed slightly better than our approach. In the case of ADP, we performed slightly better, but the most interesting result for us was to see that our approach is able to distinguish between active and inactive ligands, because we were able to reproduce the binding of GCP. You see here we got the binding of GCP with autodoc in particular, but also with addoc among the top 10 and top 11 poses, while neural plexer was unable to generate any relevant fraction. Basically, the tool generated also for GCP always conformations of the complex that have the protein in the active form, so closed, okay, while this is not the case. We also employed AlphaFold 3 for the same reason, that performed very well, but again, we were able to generate only the complexes between AMP and EDP that are overrepresented in the PDB data bank. The other ligands were not available at the time of submission. Okay, so I hope I convinced you that our approach is able to generate hololike conformations of different targets, in particular very flexible ones. I will now show you how are we proceeding in order to expand the scope of our protocol. One interesting aspect is the following. Could we exploit the simulations that we perform on one protein to get almost for free also the hololike conformations of similar proteins, and in particular proteins belonging to the same class, protein class? So by performing, for instance, homology modeling, we started testing this, investigating this, and as a proof of concept, we took again adenylate kinase in complex with a non-hydrolyzable ATP analog. The protein that we took that has this PDB code has a limited sequence identity of 45% and 64% similarity with the protein I just showed you. And in particular, the conformational changes associated to the binding occur only at the lead core interface. So the NMP domain is not involved. So it's quite challenging to see if among the conformations generated by the previous protein, there are also some that we can exploit to generate good complexes for this system. So we took all the clusters generated in the previous, for the previous system, we perform homology modeling, and we perform docking and refinement. And you see here that we were able to generate a structure that is very similar to the one found in the complex, also for this protein that is similar to the previous one, and also to rank as a second pose the native-like one. Okay, of course, now we were lucky with this system. We want to test the validity of this approach with tens and hundreds of proteins belonging to the same family. Then we also, once we assessed the flexibility at the level of the protein, we started also to assess the flexibility at the level of the ligands. Because as you know, there are ligands that are very flexible, that contains a large number of torsionals, and it's very difficult sometimes to reproduce the binding of these ligands due to the large flexibility they are endowed with. So again, we defined an approach in which we wanted to screen for conformations of the ligand that automatically fit the conformation of the protein that we are giving in input to the docking tools. Okay, so our approach is called Shaper, and in Shaper we do the following thing. We try to characterize the binding site and the ligand to help identifying native-like ligand conformation. So you take a set of conformations of the ligand, you take a protein structure where you identify the binding site, and then in output you obtain an envelope of the shape of the ligand and of the binding site. Particularly, you always get the binding site shape that is represented here with a set of red dots, okay? And you choose ligand conformations that overlap with the binding site. How we do this? We first calculate the convex hull envelope of the binding site. We refine the shape of the binding site by removing the overlap. And then we describe not only the binding site as a set of points, also the ligand as a set of points. And then we try to overlap the two, again, exploiting the alignment of the principal axis of inertia, and we calculate the volume of the intersection and the union, and we, as you see here in green, and we estimated the overlap, okay, related with respect to the volume of the ligand, in particular, intersection over the volume of the ligand. We developed a software called Gridify, and another software called Shaper. You see that here we can run this software in a Linux environment. There are a lot of options. I will not enter the details. If you're interested, you can write me. But basically, the idea is that you can define both a regular grid and also a dense grid. Okay, and you can use the connectivity between the points of the grid in order to choose the volume, the shape of the binding site in a way that you reproduce very accurately the shape of the binding site itself. So here I show you the idea on this human phospholipase A2 protein. You see, you have the shape of the binding site calculated with our approach that is represented by a transparent green surface, and you have two conformations of the ligand. One that fits very well into the binding site and has a value of the intersection almost equal to 1. One is the largest value, of course, and another one that doesn't fit very well has a very small value of this parameter. We, of course, investigated if this value was correlated with the RMST of the conformations of the ligand with respect to the true conformation assumed by the same ligand in the true experimental complex. And you can see here a graph, a scatter plot in which you have the RMST in the y-axis and the IOL on the y. Here is the experimental value of the IOL. And here you see several points. Here you see the ligand conformations after alignment to the ligand in the experimental binding site. You see that there is a good correlation below values of the RMST close to 4. You see that there is an increase in the slope. But in the real cases, we don't know the experimental conformation. So we use our approach of aligning the principal inertia axis of the ligand and of the binding site. You see that the performance is not as good as before, but still below 3 angstrom, you see a good correlation. And using this approach, we were able to retain only 10 conformations of the ligand out of 5,000 generated, okay, and use these conformations in docking. You see that this is the best pose we obtained using this approach to be compared with the best pose obtained from flexible docking. Okay, there is a slight increase in the performance because the binding pose is better, is slightly superior by using our approach. And we tested this also with a very flexible ligand, AP5, the inhibitor that I showed you before. In particular, in this case, we compared a more real setting. So the use of gEDES, site shaper, and rigid docking with 3 ligand conformations compared with gEDES and flexible docking. We use it auto-dock in this case, and you can see that if we use our approach, we sample conformations of the ligand that are as close as 2.5 angstrom with respect to the true conformation. If we use flexible docking, we are unable to retrieve native-like poses. And you can see this also by comparing the true structure, okay, that is shown in red here, with the best one generated by flexible docking. You see that the overlap is very poor, with the best structure obtained by our approach, in which we have an improvement. Okay. Finally, sorry, I will take just 5 more minutes, I guess. Now we are going to apply our protocol to perform docking and to perform in the future virtual screening on the DoD dataset. That is a very well-known dataset containing a lot of different proteins in complex with their ligands. In particular, we started with a subset, the DoD 38, in which we have a lot of proteins that have very different changes in the binding site upon ligand binding. You see that these changes are reproduced either if we take the putative binding site in our protocol that is called COACH-T. And also, if we take the experimental binding site, we have very small conformational change and very large conformational changes. Okay, at the moment, we simulated 32 proteins, and we were able to retrieve 80% of the for 80% of the targets conformation below 1.5 angstrom, and 90% of the conformations below 2 angstrom from the experimental conformation. When we go to perform docking on these targets, experimental targets, using Shaper and Gridify, and compare the results with flexible docking, we also saw in this case an increase in the percentage of native-like conformations compared to flexible docking. Okay, now we are starting to take 10 active ligands for each protein and 100 inactives to see if we are able to rank correctly the active ligands, not only on the experimental, by performing docking on the experimental protein structure, but also and moreover performing docking on the set of conformations generated by gEDES. Finally, we are set up a web server and a database. The web server, as you can see here, is not online yet. It's very still very rudimental, but basically you can upload your protein, insert your email, define the binding site, or use tools to calculate the binding site. You then submit the job, and you download a zip file that will be also sent by email, in which you have all the basically files that you need to perform gEDES simulations using the software GROMACS. Okay. And we are also setting up a database in which we will upload all the conformations for all the targets that we have been generating in these years. So you can either download them, you can download the setup, or in future, we hope to be able to provide also a full service in which we perform the simulations. So in summary, I hope I convinced you that we developed an alternative protocol to predict hollow-like protein structure, in particular for different and challenging targets. In perspective, we are going to perform several investigations. We would like also to apply the protocol to peptide protein interactions to couple it with other methods, such as co-solvent simulations, for instance. We would like to improve the clustering protocol. And in perspective, we would like to see if our data can be used to provide accurate descriptors describing the binding of ligands to proteins. Here are the references. There is also a GitHub repository where you can download all the scripts that are needed to run the simulations. There is a tutorial that explains quite in detail how to run them. Just for the sake of completeness, we applied for 2 patents that are now under review. And before leaving you, I would like to advertise the Bioexcel Summer School that will be taken in Sardinia from the 8th to the 13th of June. The registration will close next month. And among the topics, as you can see from the picture, there will be docking, MD, free energy calculations, enhanced sampling, and how to couple all these methods in BioBB. The school is mainly targeting PhD students and early postdocs, but of course, if you feel you could be interested, please apply. Then I would like to thank the people that helped me in this project, in particular Andrea Basciu, that was formerly doing the PhD and also postdoc here.
Did you stop sharing, I guess? No, you're still sharing. Okay,
good. Yeah, good. I'm done. Thank you for your attention.
So I add some link to your tutorial and to your repo in the chat. So
okay, thank you.
So
I will stop sharing.
Yeah, just some people, if they want, they have the link there. And if so, we have a question from Nikolai that was asking which software did you use for the simulation. That was exactly in the middle of your presentation. So
yeah, I'm using. We are using Gromax.
Yeah,
we are using Gromax. Yeah.
And then there was another question from Luis that is asking how people can access the web server. So I guess you gave the link for the tutorial, the link for the repo, but you spoke about a database. I guess this is what it's referring to. Do you have the link?
No, sorry. Unfortunately, I don't have the link because we started setting up this, but it's still, as I said, very, very rudimental. So we need to refine, and I hope we will be able to let's say, to set up a decent database and a decent web server by the end of this summer. Okay, we are working on it. I mean, the structure is there, but we are still working on the, let's say, ease of use, if I can say so. So I hope to have good news in the next months.
Okay, that is good. Maybe you can post on the forum when you have it.
I hope so.
Yeah. So there is a question from Christos. I will try to unmute you if you have a microphone. So if you want, you can try to speak. No, I think he has problems. I will read the question. Say thank you. It was very interesting. Did you detect a charge residue by amino acid feature and literature of, or I think is, or did you compute electrostatic for each protein structure?
No, No, we didn't compute electrostatic. We simply took the list of residues and we defined charged residues, the residue that you find in every table defined as charged ones. No, we don't calculate electrostatics. So this is interesting because, of course, there could be charged residues depending on the protein, depending on the complex that you are investigating, of course, but we didn't reach this level of sophistication yet. It could be, I mean, thank you. It's actually a very interesting suggestion. And
and there is a follow-up question. So that was, how did you calculate the binding site volume? Did you use an existing approach or did you make your own?
Okay, yes, this is a very interesting question, which is now under debate with the patent reviewer, because we claim that we use an approach that is different from the other ones. And indeed, if we compare the, let's say, the accuracy in the reproduction of in reproducing the binding site of our approach with some of the tools that are available, we see a big improvement, because we don't use in general a regular grid, but we use tight packing, so an hexagonal arrangement of points. We, as many methods, define fill void spaces, and then we define the convex hull. But then we do two things. We assign to every atom a specific van der Waals radius in order to refine the shape. And we also calculate the connectivity of each point. And we can use this connectivity to cut off, if you want, if I can say, unneeded lobes that are spurious lobes that you can find in the calculation of the shape of the binding site. So if a point is connected with less than 2 or 3 other points, all the points that are also connected to this one will be discarded. We notice that if we apply this criterion and we also took only the biggest, let me call it, cluster among all the lobes and the binding site shape that you find, we find in general very good shapes of the binding sites. Of course, this is a still matter of investigation. We tested this approach, as I said, on about 40 proteins, and we were able to reproduce accurately almost 90% of the binding sites. There are still some binding sites that are very tricky, but we are trying to work on them too. I hope I answered your point.
Yeah, I think you extensively answered. Thank you. So I think Christoph can, he confirmed. Great, thank you. So I think you extensively answered, Attilio. Thank you very much. Are there any other questions for Attilio? In the meantime, people think about, I will start sharing again because I want to take the chance to announce the next webinar. So let me see. Oh, I think sorry, I make a mistake.
If I want, I can share it, Alessandra.
No, I can share. I can share it. Sorry, I was just make a mistake in sharing the things. Yeah. So yeah, but yeah, share. No, it still not work today. Okay. So yeah, stop sharing. So I can just. I don't understand why it doesn't want to share the correct one. So sorry, I just try again. But you have the new webinar. No, you don't have the new webinar.
No.
I think, no, exactly. So I have a really problem in sharing. So, and I don't know why my
Anyway, feel free to write to ask in the website if I mean there are other questions or comments.
Yeah, that is true. So then it's also a good point. So I can put the link on the web, on the forum. So there where you can write more question if you have it. So that is exactly, so you need to log in, but that is a small things, and then it is the link to the forum where we have it. Yep, where there is the webinar of Attilio. Then we finish this section. So I thank you everybody again, and I just start to show. So that is what I want to announce is the next webinar that will be the 18th of February. And Berkes from GROMACS will tell about what is new in Bioexcel, in GROMACS 2025. And so if you are interested, please, you can enroll via the Bioexcel webpage. The registration is already open. And after this webinar, we will have another we'll start in March with 3 webinars. One is about cryo-EM, and then we have, after that, we have Elisa Fadda, and then we have Siverian Marrik with the Martini force field.
Alessandra, there is another question if we have time.
Yes, wait a moment. Yeah, so then you are so keep in track of March. So then we will have 3 webinars, one close to the other. Now I will check if there is a final question of an anonymous attendee. How do you find the best collective variable?
Yeah, of course, this is a very, very interesting question. In our case, we don't have any tool based on machine learning or deep learning to find the best variable automatically. There are many many approaches in these years that have been developed for this specific purpose. We thought that basically our approach is based on what generally we think happens during the interaction between ligands and proteins, so that the binding site change the conformation. And the approach is based on the conformational selection theory that states that if you sample long enough, even if you start from the unbound conformation of the protein, you will eventually sample also holo-like conformation. So by using our approach, we applied our approach to many proteins, and basically the fact that these collective variables are very good is the result of the basically coming from the result that we obtained. We also tested in the past, it was a long way to arrive to this one. We tested other collective variables based on structure. We tested desolvation, because in general, when you have binding of a ligand, you also have desolvation of the binding site. We tested the combination of these, also conformational changes in the whole protein, but this set of collective variables resulted to be the best one to our knowledge. Okay, so it's based on experience. There is no any theoretical approach or method that we use to say that these are the best ones. Okay, also because I mean I don't think it's so easy to state something like that.
Okay, thank you very much, Attilio. And now I will close this webinar. I thank you all the attendees attending, and for the question, and Attilio for the presentation.
Thank you very much. Goodbye to everyone.